The subject of this video is microevolution. Microevolution is distinct but related to macroevolution, the sort we have studied so far. To better understand the relationship between the two, pause the video and read this passage. This passage suggests that microevolution is just the small steps that make up macroevolution. So the changes that would lead from, say, this letter to this letter, rather than from the beginning of the passage to the end. So microevolution focuses on changes that happen over a smaller period of time. In any type of evolution, microevolution included its populations that evolve, not individuals. A population, you may recall, is a group of organisms of the same species that live in a single area, such as this picture of a population of wild boars. But when biologists look at a population, they don't just see the physical traits like this coarse hair and the tufts on the ears and how cute the little babies are. They understand that these populations have something hidden inside them. Genes. Though they're not visible to the naked eye, genes are hidden inside each of these individuals, and collectively, they talk about the combination of all of these genes as a gene pool. And inside that gene pool are the alleles that each individual possesses. Some alleles may be more common than others. For example, in this picture, it would seem that the larger bees are more frequent than the smaller bees. The fancy term for this is the allelic frequency. Allelic refers to allele, and frequency refers to how often that allele shows. For example, the allelic frequency for the large bee in this gene pool is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 out of 16, which is about 62%. Now, if biologists see populations as gene pools, then how do they see evolution? Laymen usually think that evolution is just a change in the traits of a population over generations, which is true, but microevolutionary biologists would define it slightly differently. They would say evolution is a change in allelic frequency in the gene pool of a population over time. For example, this population of beetles has a gene pool, and in that gene pool are different alleles, a green allele and a brown allele. The allelic frequency for brown is 2 out of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Until microevolution occurs when these three beetles immigrate into the population. When they do, they bring their own genes, and you can see some of those genes are brown, and so once they join the population, now the new frequency is 6 out of 18. While the frequency increased here, it can also decrease. So while this population of beetles currently has a frequency for the green allele of 1, 2, 3, 4, 5, 6, 7, 8, 9 out of 14, it probably won't be that way for long because the green beetles are not camouflaging very well and the birds are picking them off. So the allelic frequency for the green allele is probably going to decrease. And voila, there's some evolution, a change in allelic frequency in the gene pool of a population over time. However, the beetles example is oversimplified. Most traits don't show up just one way or the other, like green or brown. Most traits are polygenic, which means they display in a range of phenotypes, for example, from short to slightly taller to slightly taller to slightly taller to the tallest. If you put this range of phenotypes on the x-axis of a graph, and then count the number of individuals that have those phenotypes, most polygenic traits will show the same pattern, called a bell-shaped curve. Though it's not actually perfectly that smooth, it's more common to have an average height than it is to have a height on either extreme. So biologists often use these bell-shaped curves to represent populations, always with the same axes. Number of individuals on the y-axis, and the range of phenotypes on the x. In the height example, that would be people of the smallest sizes to people of the largest sizes in a range. Now this bell curve is supposed to represent the gene pool of a population. And in evolution, populations change. And that means that the bell curve must change too. In what ways can this bell curve change? Well, it depends on the sort of changes that are occurring in the population. And there are several types of selection that can lead to different kinds of changes. For instance, if you have a population that is staying the same over time, it is experiencing stabilizing selection. For organisms that start with one phenotype and then eventually evolve a phenotype that is altered in a specific way, directional selection is occurring. While if one species is evolving and eventually forms two separate species, something called disruptive selection is taking place. What do these processes look like in graphical form? Our first example is stabilizing selection. Stabilizing selection looks like this. If the old population used to have a bell curve shown in the dotted line, the extremes of the population are not doing very well, whereas the average individuals are doing quite well. And if the average individuals are doing well, then the population average will not change over time. An example of stabilizing selection in nature occurs in spiders. The tiniest spiders are not even big enough to get the food that they need, while the largest spiders are very conspicuous and thus likely to become food themselves. Spiders of medium sizes are the most successful, and so spider species tend to stay within the same size range. This is the sort of selection that happened to giraffe necks. While these individuals that had short necks did not survive, the ones with the longer necks did. So the average in the population started shifting towards a longer length. Lastly, disruptive selection eventually leads to the formation of two species where there was once one. If the old species had a bell curve like this, then in disruptive selection the two extremes are both highly fit. For example, with the peppered moths, the dark-colored moths survived very well in the city where the Industrial Revolution was putting soot on the trees and causing the trees to become darker in color, so they could camouflage very well there. Whereas out in the countryside where there was not any soot, the trees remained very lightly colored and the lighter-colored moths did better. The medium-colored moths could not camouflage in either environment, and so they were not as fit. So you start getting these two humps in the bell curve rather than just the one original. So remember, evolution is technically the change in allelic frequencies in the gene pool of a population. And depending on the type of selection that is occurring, you get different changes in those gene pools.